Welcome back. Today we are going to talk about a new topic, which is the cell cycle. So there are four stages of the cell cycle. There is the G1 stage, S, G2, and finally M, which is the meiotic stage, which is where cell division actually happens. The G1, S, and G2 phases are collectively called as the interphase stage. G1, S, and G2 are collectively called as the interphase. Now, moving on to the first stage of the interphase, which is G1 phase. So, in this stage, the cell prepares for the mitotic cell division by increasing transcription and translation. So, basically, the cell is synthesizing more cellular material which it needs in mitosis. Cellular materials like proteins, enzymes, cell membrane materials like lipids, cholesterol, transport proteins, etc. The cell grows in size and it must reach a specific size in order to pass the checkpoint at the end of G1 phase. And one of the proteins which the cell strives to synthesize in this stage is tubular proteins because they are necessary for spindle fiber formation, which is needed in mitosis and meiosis for the transfer of chromosomes, for the movement of chromosomes, to be more precise. And DNA is in the form of chromatin. DNA generally in the interphase is in the form of chromatin. And then it condenses later on in prophase where we enter the mitosis. So G1 is for growth. Now moving on to the S phase. So at this stage, basically DNA is replicated, and in doing so, we are doubling the genetic material. So the genetic material count goes from N to 2N, which means it doubles. And in order to replicate DNA, we will need DNA polymerase enzyme. But before that, we use specific enzymes, specific class of enzymes, which are called the helicases, which unwind the double strands of DNA, forming a replication fork, resulting in the separation of the two DNA strands. And then the RNA primers are installed at the 5' end of each strand. So we have two strands. One is called the leading strand and the other is lagging strand. So why do we need RNA primers here? This is because DNA polymerase cannot function or cannot identify DNA for replication without the help of RNA primers. So it needs RNA primers in order to identify where it should start the synthesizing of the new DNA strand from. Okay, so it acts as a marker. So the DNA strand is synthesized in the 5' to 3' direction by the DNA polymerase, and at the same time we have the DNA polymerase synthesizing the lagging strand as well, but in a discontinuous fashion, forming chunks of nucleotides, which are known as Okazaki fragments. So these Okazaki fragments have a new primer at, each Okazaki fragment has a new primer at its beginning. And these primers are made by primase enzyme. And then after the Okazaki fragments have been synthesized and leading and lagging strands are done, the primers are then removed and they are replaced by normal DNA nucleotides, and DNA polymerase does that. Then we have the DNA ligases, which stitches any breaks between the Okazaki fragments, especially. It does so by forming phosphodiester bonds. In order to prevent or to repair any errors which might happen during DNA replication, DNA polymerase has a 3' to 5' exonuclease activity, which basically removes any mismatch DNA nucleotide that has been incorrectly incorporated into the strand. And for more complex errors, we have DNA repair enzymes, which rectify any errors. So here are some graphics. So here is the origin, and then it unwinds by the helicases, forming the replication fork. And then the RNA primer, which is in red, is removed and replaced by normal DNA nucleotides. And then DNA ligase stitches the gaps. And this is for more detailed portrayal of what I explained regarding the lagging strand and Okazaki fragments formation. Now moving on to the G2 phase, which is the final stage in interphase, and it's the stage right before prophase. So if all the requirements needed for so basically at this stage, more growth happens and more transcription and translation of the genes happens in order to synthesize any remaining cellular materials which are needed for cell division. So at the end of this stage, there's a checkpoint, and the cell passes this checkpoint only if it satisfies all the requirements needed for cell division. If it does not satisfy any of those requirements, the cell unfortunately enters G0 phase. And this is how senescence happens in older ages. And this is why like the cell dies. Yeah. And it basically happens in old age. And this is why like white hair starts to appear like in the facial hair, on the beard, and on the hair. Yeah. This is the end. Please check the next video, which is going to be talking about mitosis in detail, starting with prophase. Please support my channel by liking and subscribing. I would love to hear your feedback through the comments section. And have a good day.